Hello and welcome back to Learning with Isha. I'm back with another interesting video from the Respiration series. In the last video, we've already talked about the introduction. What is cellular respiration and what are respiratory substrates? We know that these respiratory substrates are broken down in several small steps to release energy. We know that this is done in small steps so that there is less wastage of energy. Less energy is lost as heat and more of it can be stored in the form of molecules that can be utilized later. These molecules that store the energy are known as energy carriers. Why? Because they carry the energy in the cell and release it when required. The most important energy carrier is a molecule that we know as ATP. Often whenever we talk about ATP, we see that it is depicted by an explosion kind of symbol. Why is that? That is basically to denote that this ATP releases the energy in the cell. What is the full form of this molecule? ATP stands for adenosine triphosphate. Now let's talk about the very basics of who discovered ATP and when. Well, it was discovered by a scientist known as Carl Lohmann in the year 1929. But it was eventually a scientist by the name of Lippmann who showed that this molecule had high energy phosphate bonds, and the bonds, when broken, released the energy from this molecule. Now let's talk about the fact that which category of molecule does this ATP belong to? Is it a protein? Is it a carbohydrate? Is it a fat? Or is it a nucleic acid? Well, it is a nucleic acid. You'd be surprised to know that it falls in the category of DNA and RNA. This is very clear from the structure of ATP that we're going to examine now. If you look at the structure of ATP, you'd realize that there is a nitrogenous base. Besides that, there is a sugar. And on top of that, you have 3 phosphate groups. We know that this is the structure of a nucleoside triphosphate, and they are responsible for eventually forming your DNA and RNA. So now when we talk about the sugar, we know that in the case of DNA, the sugar is deoxyribose. And in the case of RNA, the sugar is ribose. In this case, we observe that both OH groups are present and therefore the sugar is ribose. So ATP is in fact closer in structure to RNA than it is to DNA. Now let's talk about which is the nitrogenous base. If you take a very logical guess, it is clear from the name itself, adenosine. So the nitrogenous base in this case is a molecule we call as adenine. So adenine is your nitrogenous base, ribose is the sugar, and we have 3 phosphate groups. Now if you look at adenine and ribose together, they are a nucleoside. So this is adenosine. Now if I consider this adenosine along with one of the phosphate groups that is still here, I'll call it adenosine plus one phosphate group. 1 kile we use the word mono. So it would be right to write that this structure is now adenosine monophosphate. Similarly, if I go ahead and include the next phosphate group also, I might as well call this structure as adenosine diphosphate, ADP. And if I also go on to include the third phosphate group, we already know that this structure is known as adenosine triphosphate. That is my ATP molecule. This is the subject of discussion today. So now we've realized that this ATP is in fact from the category of RNA. Now let's move ahead and talk about why this molecule is known as the energy currency of the cell. This is because there are 3 phosphate groups present in the structure of this molecule. And all these phosphate bonds store a lot of energy in them. They are in fact high energy bonds. They are high energy bonds. So as one of the bonds would be cleaved, a certain amount of energy is going to be released. And due to the presence of these high energy bonds, the ATP molecule serves as an energy carrier. That is why it is known as the energy currency of the cell. To understand this better, let's look at something we know as the ATP cycle. This ATP cycle is the cycle of synthesis and breakdown of ATP. So let's begin. Now you see you have an ADP molecule, adenosine diphosphate. To denote it, I'll do just what? I'll write A and make 2 phosphate groups along with it. This is my ADP molecule that is present in a cell. Now if I have to change it to the ATP form, that is the adenosine triphosphate form, it is clear that I need to add another phosphate group to this. Now where is this phosphate group going to come from? This phosphate group comes from an inorganic phosphate that is present in the cell. So the ADP along with the inorganic phosphate, they add and together form the ATP molecule. Now if you were to take a guess that is this an endothermic or an exothermic reaction, what would be your answer? Well, you would expect this to be an endothermic reaction. Why is it so? Well, this is an endothermic reaction because I already have 2 negatively charged phosphate groups present adjacent to each other. Now, if I were to add another phosphate group, I am going to introduce a lot of electrostatic repulsion. Of course, this negatively charged cloud is not going to want another negatively charged molecule to come and get added here. So I have to force the formation of this bond. To force the formation of the 3rd phosphate bond, I need to supply energy from outside. Now, where do I get this energy from? Like we spoke, we have our respiratory substrates. When they are broken down, a certain amount of energy is released. As I already said that each time a small amount of energy will be released, it will be coupled to the formation of the ATP. So we get this energy from our respiratory substrates, whether it be glucose, whether it be some other molecule. Now what we've done is we have utilized this energy and stored it. The energy is now stored in the ATP. Till when? Till the time we don't have any energy requirement in the cell. Now let's say there is a muscle that wants to contract. It requires energy. Now where is the muscle going to get energy from? From this ATP. Now what happens is in the presence of water, this 3rd phosphate bond is going to get cleaved. It is going to be broken down. Now what is going to happen is due to the breakdown of this bond, an inorganic phosphate is going to be released out. We are going to come back to our ADP, but we already know that a lot of energy is stored in this bond. Now this energy is going to get released. So now energy would be released here. In this case, the reaction will be an exothermic reaction. What is an exothermic reaction? Where a certain amount of energy is given out. Now this energy is going to be utilized by the muscle for whatever contraction it has to perform. Now you see here in this case, water is required for the cleavage of this bond. This kind of reaction is known as a hydrolysis reaction. Hydrolysis, why? Because hydro means water is causing the lysis, a breakdown of the bond. So this is a hydrolysis reaction. In the other case, it is going to be the opposite. A molecule of water is given, it is required. So in this case, it will be a dehydration synthesis reaction. This would be a dehydration synthesis reaction, because for the synthesis of this bond, a molecule of water is required. This is the ATP cycle. Now that we are aware of the ATP cycle, it would be a very logical question that might come in your mind. Energy to release हो रही है, but कितनी? How much energy is being released? Let's try to answer that question. Now let's see, there's an ATP molecule. We know that it is going to get hydrolyzed. So water, it is going to be broken down into what? Into adenosine diphosphate plus inorganic phosphate. Similarly, this adenosine diphosphate again can be hydrolyzed, and then it would be broken down to adenosine monophosphate and inorganic phosphate. Adenosine monophosphate could not be broken down any further. So we know that there are two high energy phosphate bonds. Now, how much energy is released in each step? We know this is an exothermic reaction. The energy released will be 7.3 kilocal per mole. Similarly, here also it would be 7.3 kilocal per mole. What does this actually mean? It actually means that for every mole of ATP that would be hydrolyzed to ATP, 7.3 kilocal of energy is going to be liberated. Similarly, for every mole of ADP hydrolyzed into AMP, another 7.3 kilocal per mole of energy is going to be released. Now this, mind you, is in the laboratory conditions. This is in the lab conditions. It is hypothesized that in the real life, in the actual cell, as much as 13 kilocal per mole of energy could be released. So in the actual scenario, in the cell, as much as 13 kilocal per mole of energy could be given out. Now another logical question that stems from this fact is, how much energy do you think we require in a day to keep our body going? Now you'd be surprised to know that around 100 to 150 moles of ATP are hydrolyzed every day in our body to keep us going. That's a lot of energy. Now, where is this energy utilized? It is utilized mainly by the body in ion transport. We know that the transport of molecules could be active and passive. Passive is with the concentration gradient, active is against the concentration gradient. For active transport, we know that we require ATP. Also for muscle contraction, as I've already said, nerve impulse conduction, also chemical synthesis. When you want to synthesize some molecule, you have to give energy. And substrate phosphorylation. This is something we are going to discuss in our next video on glycolysis. Now let's come to the last part of today's video, that what are the processes that release energy in the cell? That is the processes that cause ATP generation. The first important energy yielding mechanism in a cell is cellular respiration. And cellular respiration is exactly what we're talking about in this series. We're going to look at how, let's say, a glucose molecule, that is a respiratory substrate, is going to be broken down to pyruvate through glycolysis. Eventually, acetyl coenzyme A will go through the TCA cycle. Eventually, after passing through the electron transport system, we are going to get 32 molecules of ATP. Remember that this process happens in the cell in oxygenic conditions, that is in the presence of oxygen. Another mechanism that yields ATP is beta oxidation. What is beta oxidation? You see, the fatty acid chains are shortened permanently. Due to this shortening, we get a molecule that is known as acetyl coenzyme A. As we are also going to look at in this series, that this acetyl coenzyme A will further go into the TCA cycle and this is going to give us some amount of energy, some ATP. Now the third process is ketosis. Now ketosis will not be a part of our cellular respiration, so let me cover it for you. Ketosis is the process where there is a breakdown of ketone bodies in the human body. So these ketone bodies would be broken down and a certain amount of energy would be released. This process takes place in the mitochondria, and one molecule of acetoacetate, when it is broken down, it yields 22 ATP molecules and 2 GTP molecules. Now what is this GTP? GTP is another energy carrier. We said that there are several molecules that store energy. The most important one we are covering, that is ATP. Besides that, GTP is another energy carrier in the cell. These 2 GTP molecules would give me practically 2 ATP molecules. Now the last such process is anaerobic respiration. Anaerobic respiration means respiration in the absence of oxygen. So this is the only process yielding energy in those organisms that live in anoxygenic conditions, absence of oxygen. Only 2 ATP molecules would be formed here. Thus, I hope you appreciate the difference that 32 ATP molecules are formed in cellular respiration, whereas only 2 ATP molecules are given in anaerobic respiration. That's a huge difference in terms of energy. This brings us to the conclusion of this video on ATP. This video not only spoke about what ATP is, who discovered it, but also that which category of molecules it belongs to. We looked in detail at the structure, at the ATP cycle. After that, we went on to see how much energy is released in the step where we go from ATP to ADP and then ADP to AMP. We also drew a comparison between how much energy is released in the lab conditions and in the actual cell, within the actual cell. After that, we saw where this released energy would be utilized and what are the 4 different reactions or mechanisms that lead to the formation of ATP. In the next video, we're going to start talking about the first most important step of cellular respiration that we know as glycolysis. This glycolysis process is going to break down glucose and lead to the formation of 2 molecules of pyruvate. We are also going to examine how much ATP, how much energy is going to be formed in the process. If you like the video, don't forget to share it with your friends. Please press the like button and don't forget to subscribe to my channel. See you in the next video.